have heard Eileen before. She's been here really since the beginning of the center. It's been really incredibly exciting over the last year. Certainly over the years we've talked about when is there going to be something for people with chronic injuries, and Eileen and Brian have something for people with chronic injuries that's actually in clinical trial right now. This is based on their stem cell research done here at the center. So we're here at the center now over in the Sue and Bill Gross Stem Cell Center, which is just behind us. So Eileen.
Hi. Oh my goodness, in case you were falling asleep at all. I'll tell you what I've told you before in part, and then be glad to take questions. I think most of you know that my lab has two main focuses here at UCI. One is about inflammation and the role of inflammation in contributing to damage after injury. But also a big part of what we do right now is trying to understand all of what inflammation does. And it has long been thought, in terms of the inflammatory response, that it might help and hurt after different kinds of injury and disease, certainly in the central nervous system like the brain and spinal cord. So a big part of what we're working on right now is not just how inflammation might contribute to damage, but how some parts of the inflammatory response might actually be good and something that we need. So from a basic science point of view, as Jack was saying earlier, one thing that's really important to understand is that we don't, by a long shot, know everything that there is to know about the brain and spinal cord, and certainly not after trauma. And there are a lot of things that we need to understand better. So this is an example of something we need to understand better. So the other half of my lab, right, works on stem cells of various kinds. Human neural stem cells, principally ES-derived, embryonic stem cell-derived, induced pluripotent cells, or fetal-derived cells. Well, when we give a stem cell as a potential treatment into humans or into animals, we need to suppress the immune response, or those cells will die. Your body will fight them off. If we don't understand everything that the inflammatory response does, and when that happens, we may actually inhibit part of your body's potential for repair just by shutting down the wrong thing at the wrong time. So I raise that more just to, give you some food for thought and thinking about, I understand the frustration of why we do basic science alongside our translational studies. They're typically things that we really need to know because we have just gaping holes in what we understand about what goes wrong and what goes right in your body after injury. So that's half of what my lab does. Of course, many of you know the other half of my lab works on stem cells, as I mentioned. And our focus has really been to try and understand how those cells can yield recovery, what the mechanism is, whether they can yield recovery, and how the environment that we put them into affects what those cells are willing to do, how it affects what kind of cells they're willing to turn into in terms of cells that belong in your central nervous system, neurons, oligodendrocytes, astrocytes, and where those cells are willing to go, because both of those parameters really influence a lot what the potential for repair is after you put in a stem cell population. So, for example, part of Jack's research, a significant part of research in his lab, is focused on reducing the number of reactive astrocytes that are present after injury. The last thing we would want to do in terms of transplanting the cell population is add to that problem, right? So again, we have to go forward from translation and basic science hand in hand. So we need to understand the cells that we're using, how they listen to the environment that we put them in, whether we can understand the molecular signals that control them, right? So that potentially we can transplant cells and maybe a particular kind of drug or molecule at the same time to say, no, don't become an astrocyte, right? So that we don't get both an improvement and some things that may inhibit the potential for repair at the same time. Having said that, my lab has been very focused on trying to focus on the more delayed response to injury and whether we can transplant, in the case of stem cells, into more chronic injuries. And there are a couple reasons for that were touched on a little bit a minute ago, but I'm going to reiterate them just in part. One, there are an awful lot of people that are living chronically in the United States with spinal cord injury, right? The St. Christopher and Dana Reeve Foundation survey with the CDC several years ago identified 1.3 million people in the US living chronically with spinal cord injury, right? And that's important for us to understand because from a dollar to benefit point of view, having a significant population that we can identify matters in terms of raising money. It matters in terms of even how the FDA looks at the crisis that we need to be able to go forward with. That means we need to think about developing translational work, developing therapies that will target that population. And those are really hard experiments to do in the lab, but for that reason, that's where we predominantly focus. The other issue that matters quite a bit is the level of injury, right? So we have people here in this room that are focusing all on different levels of injury. So Leif in the very low part of the spinal cord. Most folks work in the mid-thoracic region. It's relatively easy to do those surgeries, to work in animal models that we're all dependent upon to get our preliminary proof of concept to be able to go forward. But very, very important to think about cervical injuries, right? For several reasons. One, it's the majority of injuries in the United States. And two, we can, with a therapy that yields relatively little repair, we might be able to get a segment of function back, right? So this much in terms of distance. Jack gave a basic science talk yesterday here where he made this point very eloquently. And it's really important to consider. The amount of regeneration, the amount of growth past that injury that we need is relatively small. The number of cells that we might need to turn into myelinating cells and repair myelin that's damaged is relatively small if we're just talking about getting this versus that, right? So that's a big focus for us. So on the basic and translational side of lab right now, our very significant focus is not just understanding these microenvironments and how they control what stem cells do, but looking towards cervical models and eventually moving into cervical spinal cord injury trials if we can get there. Okay, so I'll just go ahead and say a couple of words. Many of you, again, have heard this before. The work that Brian Cummings and I, and of course, many of the people in my lab over the past 10 years have worked on with a fetal-derived cell population, human neural stem cell population, in collaboration with Nobuko Uchida and a company called Stem Cells Inc. up in Palo Alto, is now in clinical trial. Stem Cells Inc., and we agreed, chose for that trial to do a very small single-site focused trial, not a multi-center, multi-site. For that reason, they chose to go forward in Switzerland at the University of Zurich Balderst Hospital, so that's where the trial is ongoing. That trial initiated in December of 2010. Approval was granted. Enrollment opened in March of 2011. And the first cohort of patients was completed for enrollment and transplanted in December of 2011. So 3 months ago, basically. So let me just say a couple of quick words about that trial. It is, I think, different in probably 3 ways than most trials that have gone on so far. One is that it focuses on this slightly more chronic period. So focusing on individuals that had spinal cord injuries 3 to 12 months prior to transplant, right? So you can't enroll in the trial unless you're at least 3 to 12 months out. There's a couple of reasons for that. One is in terms of being able to really recruit people into the trial who are at a better place to understand what they may be getting into in terms of risks. 2, because it may be, and certainly the hope in the field is that by moving into a more chronic injury situation, we will be able to use smaller numbers of people in an efficacy trial to be able to see if there's recovery. And the reason for that is, in the short term after injury, there are people that recover spontaneous function. And the amount of that recovery decreases over time. So by waiting at least 3 months for enrollment, or preferably 6 months for enrollment, you can take what would have required 600 patients to be able to determine efficacy for a phase 2 trial and get down, for a cervical trial, for example, to as few as potentially 25 patients. Well, that has a big impact in terms of how much that trial is going to cost, whether you can convince venture capital folks to go along with funding a trial like that, and whether the company that you're working with is going to hit this valley of death, which is a very critical issue that Jack raised. So that's one way this trial is different. Of course, you have to have the preliminary data to show regulatory agencies that you have efficacy in that period. So you have to build all of your studies around looking at the timeline of when you can get things to work versus not. So that's one way it's different. The second way that it's different is this is actually a phase 1/2 trial. So that means it looks at first safety, first and foremost safety, that's our job. But second of all, it has, we hope, the potential to be able to get at some efficacy measures, and that's because it's into a more delayed patient population. So the next cohort, I mentioned that the first cohort of patients completed transplants in December. That was 3 patients that were thoracic Asia A. The next cohort of patients, we have to wait 6 months and accrue safety data on everyone involved. The next cohort of patients will be Asia B, and the cohort after that will be Asia C, until we get to a total of 12 patients enrolled. So Asia B means that there's some sparing of sensory function. Asia C means that there's some sparing of sensory and motor function, both. We'll see. The hope is that by being able to get approval to move into Bs and Cs and not strictly within the Asia A population, we may have a shot to be able to get some efficacy measures. But we won't know until 2 or even 4 years down the road in terms of how the enrollment goes. The third way that this trial is a little bit different, and I think this is kind of important to mention because certainly people call my office all the time trying to evaluate what's out there for potential trials, is that the cell dose in this trial is a therapeutic cell dose. So what do I mean by that? I'll use the Geron trial as an example. The Geron trial transplanted into humans the same dose of cells that was used in rats. Your spinal cord is not the same size as a rat's spinal cord. So that's the approval based on the safety data that they were able to get for transplantation. But it leaves a little bit of wild card open for whether or not that small number of cells will be able to scale up and yield repair, right? So in the stem cell link trial, it was possible to get approval because there's an extensive safety profile for these cells already in humans to do what's called allometric scaling. So that means we take the dose that we used in our rat and mouse efficacy and safety studies, and we recalculate that based on the size of the human spinal cord. So the number of cells transplanted in the mouse, for example, when we do our experiments, is just 75,000. The number of cells that's transplanted in the case of the stem cell link trial in Zurich is 20 million. And that's just straight allometric scaling for what we know in our animal models based on safety and efficacy data. So that's a quick summary of the stem cell link trial. We really won't have anything to report. There have been no adverse events, as there have been no adverse events with the Geron trial up until its halt. And if all continues to go well, we hope to begin enrolling the next cohort in June or July of this year.